A park, a place of tranquility, and an oasis of life. A walk through the park is a simple pleasure, a moment to breathe, to listen, to observe. The laughter of children, the rhythmic footfalls of joggers, the sound of people exercising. An endangered species, the Komodo dragon, a large reptile of the monitor lizard family, Varanidae, that is endemic to the Indonesian islands, can be seen gliding across the pond. But beneath this familiar world, hidden just beyond our sight, another universe exists.
Don't you ever wonder what's in the water? Not just the stuff we normally see, but the smallest stuff like microorganisms. A single droplet of this water, barely noticed by those passing by, holds more life than the entire park itself. Millions of microorganism creatures, so small they exist in a world beyond our vision, shaping this ecosystem in ways we are only beginning to understand. Well, today, we'll be investigating microorganisms in water ecosystems. So come join us as we collect our 5 different water samples.
Our first location, the Mangkut Ramayasalan Park in Nonthaburi. This isn't just a place where you can come exercise, but also a lovely place for us to collect some water samples in the large pond here. We use this water collecting to borrow from another group to help us with this process.
For centuries, the Chao Phraya has been the lifeblood of Thailand. A single drop from the Chao Phraya holds a vast and complex ecosystem, where microorganisms work tirelessly to sustain the river's delicate balance. These microscopic life forms, plankton, bacteria, algae, and are the foundation of the river's ecosystem. Some provide oxygen, others filter pollutants, and all of them play a role in keeping these waters alive. Yet, the balance is fragile, which means that this place is another lovely place to collect our water samples.
A fish tank is a world in miniature, a delicate balance of water, life, and unseen forces. To the casual observer, it is home to colorful fish and flowing plants, a peaceful microcosm of nature. But beyond what we can see, an invisible community is at work, shaping and sustaining the world. To the fish, this world is home. To us, it is a mystery until we look closer.
An ice rink is a world of motion and grace, a place where skaters glide effortlessly, their blades whistling against the frozen surface. But beneath this smooth, glittering sheet of ice lies a world few ever consider, a world of hidden life, waiting for its moment to return. This ice, once flowing water, was home to an invisible ecosystem, countless microorganisms trapped in suspended animation, waiting for warmth to awaken them once more.
A quick stop at the dispenser, a sip between classes, cool, refreshing, and clean. But within every drop, an unseen world exists. Harmless bacteria drift in the current, or resilient biofilms, the layers of microscopic life form on the surfaces within the pipes. Some microorganisms thrive even in the cleanest water, clinging to the very infrastructure that delivers our daily drink. Others arrive from the air, from our hands, from the world around us, too small to see but always present.
Hello guys, so we just finished getting the water from our last sample, the school water dispenser. So we just want to talk about the hypothesis for this project. I feel like the pond water will have the most diversity of microorganisms.
Yeah, because it's in the nature and no one goes to clean it or anything.
Yeah. And what's next? The Chao Phraya River, right? Yeah, because it's a river, so there should be some living things in it, I hope. And next is Mo's fish tank. I'm actually not sure about this one because she has a water filter, so
so it should be pretty clean.
Yeah, unless her fish poops a lot or whatever. And then the fourth one is the ice rink. I'm actually not sure with this one as well, but we chose it because it's an interesting pick.
Yes, but I'm pretty sure it should have some bacteria or something in it because it's dirty.
Yeah, it really is dirty. And then lastly is the school water dispenser, which we just got. So what do you think?
It should be clean. There should be no bacteria at all because it's supposed to be drinkable and we're already drinking
Exactly.
it.
Yeah, so that's our hypothesis. So see you after the lab. Goodbye.
The results of this experiment is that we discovered the most microorganisms in the water from the Mayasan Park's pond. The first picture you see here could be a type of diatom, a single-celled algae with a silica shell, or a dinoflagellate, a microscopic plankton with a protective algae covering. The second one, it's likely cyanobacteria or algae filaments. These filaments might also be fungal hyphae, which are part of fungal networks involved in decomposition. The third has a possibility to be, first, bacteria, cocci or microcolonies. If the water sample is from a pond, lake, or river, these could be spherical bacteria like Micrococcus or Staphylococcus forming clusters. Some bacteria also form biofilms, which might explain their scattered appearance. Second, protozoa, cysts or small flagellates. Certain protozoa like Cryptosporidium or Giardia can appear as small, rounded cells under the microscope. Some free-living flagellates also have round resting stages. Third, algae, chlorella or other small phytoplankton. If the water is rich in nutrients, these might be a single-celled green algae like chlorella or Anchistrotismus, both of which are common in freshwater ecosystems. Fourth, yeast or fungal spores. Some fungi release spores into water, and yeast, like microorganisms, can also be present, especially in water with decaying organic matter.
For the fourth one, we use methylene blue, and we believe that this could be 1. Bacteria, gram stain or biofilm fragments. If this sample was stained, the darker colored clusters could be colonies of bacteria, possibly gram-positive bacteria. This scattered formation may indicate bacterial biofilms breaking apart. 2. Protozoa, encysted forms or trophozoites. Some of the larger circular shapes could be protozoan cysts or free-swimming stages of organisms like amoeba or paramecium. 3. Algae or cyanobacteria. The darker blue and greenish areas could indicate the presence of cyanobacteria or microalgae, which thrive in freshwater environments. 4. Fungal spores or organic debris. The irregular clumps at the bottom might be fungal elements, decaying organic matter or contaminants in the sample. We also use methylene blue for the fifth one. The star-like structure visible in the microscope image strongly resembles a desmid or Asterionella, a type of diatom, both of which are commonly found in freshwater environments. Here are the possible identifications. First is Asterionella diatom, a clonal diatom species that forms star-like or chain-like arrangement. Diatoms have silica-based cell walls and are crucial primary producers in aquatic ecosystems. Second are desmid, green algae, order Desmidiales, a group of unicellular green algae with symmetrical and often elaborate shapes. Desmid thrive in nutrient-poor freshwater and are known for their geometric appearances. Third, phosphor or protozoan structure. Some protozoans or microscopic invertebrates may have spiky extensions or ciliated appendages that create a star-like silhouette. Lastly, microscopic image of a clustered organism, possibly fungal or bacterial colony, algae, protozoa, or small multicellular organism, brownish-reddish color.
Overall, the experiment was a success even though we hope that we might have been able to discover more microorganisms in the other sample of water as well. The results show that one location can have many microorganisms living together and that the identification of bacteria, protozoa, and algae provide insight into the ecological health of the water bodies by the presence of potentially harmful bacteria in certain samples indicated possible contamination, emphasizing the importance of monitoring water quality for environmental and public health purposes.